Assalamualaikum everyone. In today's lecture, we are going to discuss about the introduction and the history and the scope of microbiology. So this is the first lecture of microbiology. So before we are going to start the lecture, I request you to please subscribe to my channel, like my videos, and also press the bell icon to get the notification of all the videos which you have seen and all the videos which you don't see. So now let's start the lecture. Bismillahirrahmanirrahim. So microbiology, first we are going to define that the microbiology is the branch of biology that deals with the study of microorganism, and the microorganism can be called as microbes. Okay, so these microbes are actually microorganism, and the branch in which we are dealing with these microorganisms, this branch is called as what? Microbiology. So this was the definition, and now let's see what is the microorganism. Microorganism is only composed of one cell. They can be unicellular or multicellular organism. They are distributed in both, so they are present where is the presence of microorganisms. So they can be present in the bodies of plants, in the bodies of animals, in air, in water, in soil, in marine, almost everywhere on the earth. So the microorganism is present everywhere. You cannot locate a fixed position of them. Now let's see what is the size of microorganism. They are usually less than 1 millimeter in diameter, and we use microscopes to see them because they have very small diameter. There are some examples of microorganism written here. For example, virus, bacteria, fungi, algae, and protozoas. They are all included under the microorganism category. Now let's see that what are the techniques for the separation and isolation of microorganisms. So uh, some organism studies the microorganisms. Sorry for this. Yes, some microorganism studies by the microbiologist can be visualized without the aid of amplifications or microscope. So they are going to say that some biologists suggest that some types of microorganism can be seen with the naked eye, okay, without the help of any microscope. So then we consider them microorganism. Yes, we consider them. For example, fungi, you can see the fungus floating over the water which stands for many months and days. So some types of microorganism can be seen with the naked eye and some cannot be seen. So there is no specification that always the microorganism can be seen under the microscope. Okay, they can be visualized, but mostly a microscope are seen under the microscopes. So there are some techniques written here to isolate and to culture the microorganism. What is mean by culturing? Culturing mean to produce the microorganisms or to culture and develop and nourish them in the laboratories. Okay, so we have to do this because we need some types of desired product from them. So in order to isolate, we done three processes. We done isolation, we done sterilization. The process of sterilization is to prevent the germs which are present on the instrument. So we have to sterilize the instrument before the use and before performing any practical. And last is the culture in artificial media. We provide some type of nutritions to these microorganisms and prepare them artificially in a lab. Next is that a microbiologist may be interested in specific type of microorganisms. So actually, these are some branches of microbiology. This is virology. The virology is a branch of microbiology in which we are dealing and studying about viruses. In bacteriology, we only study about bacteria, their types, their property, and everything. In phycology, we always study about algae. In mycology, we always study about fungi, and in protozoology we always study about protozoas. ठीक है. So in there are some more branches of microbiology written here. For example, medicinal microbiology, and the immunology is considered under a type of microbiology, which is medicinal microbiology. Second is the food and dairy microbiology. We discuss with the presence of microorganisms present in the food. Second is the public health microbiology. It can also be called as epidemiology. Then there is industrial microbiology, and last is agricultural microbiology. So now let's discuss the branches of pharmacology, microbiology one by one. So first branch is the pharmaceutical microbiology. So pharmaceutical, as its word indicate, that there is application of microbiology to the pharmaceutical and healthcare environments. So pharmaceuticals means we have to prepare some kind of dosage forms. Okay, so in order to prepare dosage, we need to learn about the microbiology, because if we are unaware about these microorganisms, we cannot prepare a pure drug. So there is possibility that these kind of microorganism can enter into the drug, so therefore we have to prevent them, or in order to prevent them we use a term which is called as pharmaceutical microbiology. So there is risk in both proactive and reactive to gathering in the testing material, because whenever you are going to test a material and perform various kind of tests on your drugs, there is possibility that some kind of germs can enter, and therefore you should have to adopt a monitor environment in order to prepare a specific drug. There are some characteristics of these microorganisms. For example, microbial morphology. In this branch, we see study about the structure of these microorganisms. We study what types of bacteria are present, what shapes of bacteria are, what is the shape of algae, what is the shape of fungi, and all of these. So we also study about the microbial histology. We also study about their cells and their morphology. We also study about the physiology, that how they enter into the living organism and how they undergo their life cycle. Then we study ecology, that how they impact and affect the environment. Then we study microbial genetics, in which we study what is the type of genetic material which is present in these microorganisms. We see molecular biology, in which we see what types of molecules are present in them. Then we see microbial taxonomy. In this, we divide the bacteria into their class, subclass, and kingdom, and such like that. Okay. Now we are going to study about the histological preservatives. Yani we are going to discuss the history of microbiology. So, whenever we are going to discuss the history of microbiology, always a scientist name came, which is the Robert Hooke. So, Robert Hooke prepared a crude microscope in 1665. He made a crude and the first microscope. And when the Robert Hooke see in this crude microscope, he sees small, less structured unit, which he called as cells. So, Robert Hooke made the first discovery of a cell. Then the second historical event made by Antony Leeuwenhoek. This scientist observed first living microbe. So he used his single length magnified microscope, and with the magnification of 50 to 300x magnification. So the words which are written in this highlighted part are not clear to you, but you can retain it. The words written here are 50 to 300x magnification. So between 1674 to 1723, he wrote series of papers describing his observation, that he observed bacteria, he observed algae, he observed protozoa, and he observed fungi and animalcules. He called them animalcules. Okay, he doesn't call a bacteria, a fungi, a algae. He collectively call them animalcules. Okay. Here you can see the picture of her his microscope. This is the lens of his microscope, in which he sees specimen holder, and there is the focus screw handle, and after that he sees it. And here you can see how they see. And this kind of picture he see under the microscope, some types of this shape, some this shapes, some that like shapes he observed. And he see and call all of them as animalcules. Now next we are going to discuss spontaneous generation. So, spontaneous generation is just a theory which is given and it is rejected. So, there is a belief, early people believe that there are some types of vital forces present in non living organism or decomposed matter, and these non living material undergo and produce a living material. So, this process is called as abiogenesis. Okay, so, in other words, if we clearly define abiogenesis, so it is defined as organisms arise from non-living material. Mean living organism is arising from non-living organism due to presence of some vital forces in non-living or decomposed matter. So this is called as spontaneous generation. Now, next scientist is Louis Jabot. So, in 1670, he conducted an experiment in which he divided a hay confusion that boiled into the two container, and he heated container with the closed air, and other container is freely open. Okay, he take two container, number one container and number two container. Okay, so this was the number one container and this was the number two container. So first container he covered it, closed its end. Okay, while the second container is being opened. Now he heat them. Okay, he give the heat both of them, and after heating he see that open container develop microorganism. And so therefore he disapprove the process of abiogenesis. Okay, now, the second experiment performed by two scientists, Reddy's and Japlots. So, Reddy's experiment is that he placed a meat inside a container, and he opened one container is closed, other is open. So he see that the closed container in which meat has no type of any microorganism, and the container which is being opened, and then he see some kind of flies hatching on it. Then the Jacob experiment, he is same. The diagram is explained, which I have drawn in the previous slide. Next discovery is made by Louis Pasteur. You know very well, Louis Pasteur has a great role. So he disapproved spontaneous generation of microbes by preventing this particle from reaching the sterilized part. Okay, or in 1861, he completed his experiment later to rest the spontaneous generation. He showed that microbes cause fermentation and spoilage. So this was the experiment which is performed by the Pasteur, as you can see here. Um, in the first part, he take a vessel like that, and he put some type of fluid in it, and the fumes is rising above in this tube, and like goes to like that. Now what he do, he put some types of bacteria in this part of the tube, and after that the bacteria and dust in air settle in this band. Then after that he tilted this vessel like this, and due to this inversion, these bacteria enter into these flasks, and after the entrance, after many hours and days, these microbes enter, react with this solution and cause its spoilage. So after this, these bacteria goes to multiply, and they uh, spoil that solution. So in that way, the pastor rejected that some non-living is producing living material. So he rejected that. He said that there is no possibility of abiogenesis. So now we see what is the role of microorganism in producing the diseases. So first, a scientist who demonstrate that is the Olive Holmes. Olive Holmes showed that sepsis, this is a type of disease, and he show why this disease is going to transmitting. So he say it could be transmitted by the hands of medical students and may cause the disease. So he say that due to because in early days medical students don't sterilize their hands, So due to this, the disease from one person can be transferred to a healthy person. So he show why this disease is going to be transmitted. Second scientist is M.J. Barclay, and he say he great potato blight of Ireland. Ireland is a place where the potato is going to be spoiled, and the people don't know why they are spoiling. Then this scientist caused and find out, and he observed that fungus is the cause of that disease. So last scientist is Louis Pasteur, and he show that the disease of silkworm is due to a parasite protozoan. So in that way, there is a proof that these microorganisms are the cause for causing a disease. So, in order to recover from this disease, Edward Jenner was the first scientist who make first vaccine, and he make first vaccine against the smallpox. And Louis Pasteur developed other vaccine for the other diseases like chicken cholera, anthrax, and rabies. Now again, it is revised that in 1796, Edward Jenner made first vaccine against the smallpox and he injected the weakened and the then antigen of smallpox, and he injected that into a small boy, and after that he see that some types of symptoms appear in him, and after that he live as a healthy person. So he concluded that this is the process of vaccination by which you can treat a disease. So there is again a history that in 1887 vaccine against the rabies is generated by Louis Pasteur. And Robert Koch, he developed a criteria by his teacher Jacob Henle, and he established there is a relation between the bacillus and anthrax. So bacillus anthrax is a bacteria which cause anthrax. So he is think that there is a link between a particular microorganism and a particular disease. He create the idea that the microorganism which causing a disease, there is a link between that microorganism and this disease. So Robert Koch confirmed that there is a presence of germs. So history is that in 1884, Robert Koch produced postulates, and he see that how there is a transmission of diseases. So postulates of Robert Koch are: the causative agent must be present in all affected organisms but absent in healthy individual. So he say whenever the person is affected, any kind of causative agent or any kind of germ will be present in her, and if and in a healthy person this causative agent will not be present. This agent which is responsible for producing the disease can be isolated and cultured. Third postulate is that the cultured agent can be introduced into a living organism and same disease can occur. Fourth postulate is that same causative agent can be isolated from affected host. So these are some postulates of Koch. Now there is a development of culture media, that how we are going to develop this culture media. So there is a possibility of isolation of pure culture of only one type of organism, especially important during the coach period. Gelatin cannot be used for solidifying material. Okay, so for in order to prepare a culture media we need a gelatin, but we cannot use it because gelatin melt at 28 degrees centigrade or less than it. So some bacteria can hydrolyze this gelatin by the enzyme. So his wife, coach wife, his assistant, she proposed that we should use agar instead of gelatin, because agar cannot be digested by most of bacteria, and second thing, it melts at 100 degrees centigrade, and it can be used only 2%. Now another assistant of coach is Richard Petrie, and he introduced the Petrie dish. Last is the development of vaccines and anti-sera. Edwin Jenner was responsible for it. He introduced the vaccine first for the cowpox, and he see that cowpox and smallpox are interrelated, and he called whole of these procedure to vaccination. Now, vaccination. What is vaccination? Vaccination is the inoculation of healthy individual with a weakened form of microorganism. We prepare a weakened form of microorganism and inject it in a healthy individual, and same we can protect from this disease by the production of antibodies. Other is the posture and Roberts incubating longer prior Nangazen, and he said Pasteur and Roberts reported that some kind of, if we see if we kept the culture for long time in the lab, they can cause the production of bacteria in them. So this was seen by Poston and Robert working with chicken cora, and they noticed that animal injected with intended culture were be resistant to the disease. So if we kept them in long time, they will become resistant to a particular disease. So this was all about today's lecture. I hope that you will enjoy this lecture. If you like it, then subscribe to my channel, like my videos, and also press the bell icon to get the notification of all the videos which I have uploaded further. So thank you so much for subscribing me. Take care of yourself. Allah Hafiz.